Cadmium, chromium, lead and nickel are a number of toxic heavy metals that persist in the soils due to mining activities, agricultural fertilisers and metal refining. These metals are toxic to the environment and quickly absorb into living life, like Jimmy right here. He is one of thousands worldwide who have experienced heavy metal poisoning. Heavy metal contaminated soil can be cleaned chemically, but it is expensive and the precipitants are difficult to remove. Sorry, Jimmy. Phytoremediation offers an economically environmentally friendly alternative to remedy the soil to allow the return of natural flora and fauna. But what is it and how did it get to where it has today and is implemented? Welcome to the genetic basis of stuff and things. Phytoremediation is an off-branch from the pioneering work by Jackson, Simmons and Berg, who were the first to genetically modify organisms. In 1972, these scientists edited viral DNA to allow for the first time in history the transformation of target DNA to cells of interest. This caused a spark of controversy and the shutdown of all gene-related research for 2 years. However, through its innovation, the gem of phytoremediation was born. Phytoremediation, coined in 1983 by R.L. Chaney in their paper on zinc accumulation in Vigna, unguiculata, is the process of utilising and often genetically enhancing plants' natural ability to accumulate heavy metals in the soil, effectively decontaminating it. Toxic heavy metals such as lead and cadmium are dangerous because they displace essential metal ions and can cause disrupted release of neurotransmitters, impaired DNA repair, and lead to death. In today's grand knowledge, we now understand the primary mechanistic flow in phytoremediation. It is facilitated by root exudate that changes the pH of the rhizosphere to increase the solubility of heavy metals in the soil. These solubilized metals enter root cells via the symplastic and apoplasmic pathways where they are transported to and stored in root cell vacuoles or travel to the xylem to be distributed and stored in the vacuoles of all the cells of the plant. In the case of cadmium, once inside the cell, glutathione S-transferase forms a metal complex by catalyzing coordination bonds between the metal ion and a thiol group with 2 glutathiones. This bond is fairly stable, however, to ensure the metal ion cannot disrupt major metabolic processes, yeast cadmium factor protein 1, an ATP binding cassette transporter located in the clonoplasm, discovered by Shipka in 1994, and Saccharomyces cerevisiae, transports glutathione conjugates to the vacuole where they are stored indefinitely. Despite lead being one of the leading heavy metal contaminants, any genes relating to its accumulation eluded scientists in the late 90s. In 1998, Raising discovered the first lead resistance gene in bacteria. However, the gene was an ATP pump and conferred resistance by pumping out absorbed lead, so it was unsuitable for lead phytoremediation as it was not accumulated intracellularly. Years later, Song realized the answer was beneath his nose the entire time. Song hypothesized that the same mechanisms employed for cadmium transport to the vacuole in yeast could be used to detoxify and accumulate lead, as it is known to form complexes with thiol compounds and therefore could likely conjugate to glutathione. Song created mutant Saccharomyces cerevisiae with a dysfunctional yeast cadmium factor gene and compared their growth to wild-type strains on both cadmium and lead agarose gel plates. In both settings, the mutant yeast experienced suppressed growth. Following, the team performed a recovery experiment where a plasmid containing the functional gene was uptaken by the mutant strain, restoring the mutant strain to wild-type phenotype. To confirm suspicions, Song duplicated the yeast cadmium factor gene using PCR and inserted it into a plasmid with 4 enhancers to overexpress the gene. Following, the plasmid was transferred to a bacterial species, Agrobacterium tumefaciens. The bacterium was used to infect Arabidopsis thaliana cells, transferring the plasmid, allowing the overexpression of the gene. The plants overexpressing the resistance gene were significantly larger and healthier than the wild types, but they did not accumulate a high concentration of heavy metals on a per-weight basis. This was somewhat expected as YCF1 is a transport protein located on the tonoplast and therefore has no control over the uptake of the heavy metals by the root cells. Instead, it enhanced the growth of the plants by minimizing time the glutathione conjugates spent in the cytoplasm, subsequently minimizing the metabolic impedance caused by the heavy metals. However, ironically, by the time of publication, Song was not the first to have discovered a lead-accumulating gene. A few months earlier, a Spanish team led by Gisbert identified a different gene that conferred lead resistance and experienced similar results when they transformed their gene using Agrobacterium internictana glauca. Together, these scientists showed that lead-accumulating genes exist and more may be hiding within the genes already discovered. However, you still may be asking yourself, is phytoremediation used today? Scientists have identified numerous more genes following the pioneering work of Song and Gisbert that cause phytoremediation plants to grow faster, accumulate more, and absorb a greater variety of metals. Despite these advances, challenges still remain. Phytoremediation plants are expensive to produce with the gene editing required to optimize their accumulation capabilities. The accumulated metals are often stored in the leaves of the plants, causing heavy metal poisoning when ingested by wildlife. Moreover, releasing genetically modified organisms could have unforeseen consequences on the ecosystems. Phytoremediation is an exciting avenue in genetic engineering, but challenges still exist before it is ready for use. However, without the hard work of scientists like Schipka, Gisbert, and Song, phytoremediation will not be the promising solution to heavy metal soil contamination that it is today.